Hello.
Hi.
Hi, how are you? Hello, Jesse and Mehul. Yeah. All right, let's get started. So, this is first meeting in a few weeks. We had some things we haven't been able to have a meeting. We've been doing our open source meetings, which are on Fridays, and we've been doing it's kind of a different environment there, where we're talking about different open source projects, different issues in open source. Last last Friday, we talked about open source in education and ed tech, some open source projects that are ongoing and how people, um, work through them and how they change things and how they kind of work through things like agentive coding. The meeting on Fridays is at noon Eastern time, so it would be like about 2 hours ahead of when this meeting starts on Fridays. So that was for Mehul. Okay. Yeah, Morgan is here. Hello, Morgan. So I have an announcement from the OpenWorm Foundation, and that is John White, who was a pioneer in connectomics and in working on microscopy and things like that. He donated his Wiley Share Prize to OpenWorm. So this is a major sort of milestone in the OpenWorm Foundation, where we've had to raise money in the past, and that's been done through crowdsourcing or other types of means. A lot of times it relies on small donations, but this, I guess, is a larger donation where John White, who's been part of our scientific advisory board, has donated uh, prize money to the effort. So this is the Tumblr blog post for OpenWorm. OpenWorm has a Tumblr account, and so this is their announcement. So in 1986, John White and colleagues published the structure of the nervous system of the nematode Caenorhabditis elegans, a paper universally known as the mind of a worm. It was the first complete wiring diagram of any animal's brain, 302 neurons and more than 7,000 synapses, mapped by hand over years of painstaking work at the electron microscope in Cambridge. This is the lab that Sidney Brenner started, and in addition to John White, of course, Sulston was also there, John Sulston, and he did a lot of the electron microscopy for development for the embryo. So when you see embryo images of when we have a C. elegans embryogenesis image, when you see that kind of microscopy, that's something that was pioneered by John Sulston. When you see anything referencing the C. elegans connectome, that was pioneered by John White. And of course, when they did this back in the 70s and 80s, they had to do this using non automated techniques. So a lot of times it was, drawing things out by hand. They didn't have the benefit of tracking software or anything else. So it was quite an undertaking at the time. And of course, they could work these things out. They could work out embryogenesis, different stages of embryogenesis, the positions of cells, the nomenclature of cells, and so forth. And they could also do things like look at how neurons are connected and then identifying synapses and images. So this is a little bit of technical insider talk, but basically the way they would do a connectome is they would take microscopy images of the neurons and they would look for these things called gap junctions. And the gap junctions are where two cells meet and there's a passageway between them or a pore. And those pores are the gap junctions. And those are called electrical connections. And they form the electrical connectome. So they mapped out the gap junctions between cells, and then those would count as the edges of a graph, where the cell bodies being the nodes of a graph. So you'd have this network that would form from that. So every cell was a node, every gap junction was an edge, and you had your basic connectome. And then, of course, people would refine the techniques after that. People used better microscopy, used automated techniques, and so forth. And people have also developed connectomes or techniques to look at connectomes in defined mutants in males. So the default in C. elegans is the hermaphrodite. There's also a male connectome, and of course, you know, there are different drafts of the connectome where people have kind of defined different connections in different ways. So when we talk about synapses in the C. elegans connectome, we're talking about those chemical connections, and so cells uh, have synapses, which are where they send out axonal processes, and at the end of those axonal processes, you have synapses or little what they call boutons, which communicate with each other, and those synapses then transmit chemical signals across the gap between them. So that's a different form of communication, and it requires a different type of sort of identification because you have to look for these little boutons throughout the image, and you have to count them, and that's a painstaking activity. And of course, if you have better resolution microscopy, you can do a better job of that. You can figure out through other techniques like staining or other types of markers what kinds of what kinds of synapses they are. Maybe they're sensitive to certain chemicals over others, and so forth. So that's kind of how we started with connectomics in C. elegans, and really connectomics more generally, because C. elegans is a model for connectomics for very small connectomes. And then, that's something we can define pretty completely. And then we can maybe move on, and as we have, to Drosophila connectomes, which are bigger and more complex. So this is really kind of where it started. So that paper that I talked about with respect to John Waite, that was the foundation on which OpenWorm was built. So OpenWorm started as a sort of the mission was to simulate or emulate the C. elegans nematode. And one of the core parts of that is emulating that connectome and using the data sets that were generated in part by John Waite and by John Sulston to reconstruct the worm. So that's where this connection comes from, very deep connection. And so John Waite won the Wiley Prize in Biomedical Science, or I guess he was a partial recipient, and then he donated his share to the OpenWorm Foundation. It's the largest single gift in the history of OpenWorm, and this is a little bit about John Waite, celebrating John. So John was awarded the Wiley Prize alongside doctors Rubin, Murthy, and Seung for foundational contributions to understanding neural circuits via connectomes. The prize ceremony took place at Rockefeller University on April 10th of this year. Congratulations to John on this recognition of a body of work that has shaped an entire field of science. And so this is a vote of confidence that, in providing this gift, that he thinks that there's still potential in OpenWorm, that there's always potential in OpenWorm, and that we can do great things. And so this is kind of drawing from the roadmap of the executive and the science advisory committees. So what we're building for 2026, the sort of the organizational push in OpenWorm as a whole is to develop three things. The first are design documents, which are of course the sort of the documentation of the project to guide people. So 29 draft design documents are currently under scientific review. They serve two purposes. The first is preserving institutional knowledge so newcomers can learn by reading rather than by finding a mentor, and then providing concrete implementation targets that break down into well scoped contributions. Um, this is inspired by RFC processes used in Rust, Python, and Apache. They formalize decisions the project has already made and create a transparent process for proposing new ones. The second push is a contributor progression model, so a clear pathway from newcomer to core contributor, drawn from the open source projects that has successfully scaled communities well beyond their founding teams. And then the third one is AI assisted contribution, which are pathways that use AI to lower barriers to participation, included automating onboarding, AI augmented code review, and a model where contributors can direct AI agents to work on well scoped issues defined by the design documents. The science is human led, every specification is human authored, every merge is human approved. AI provides extra capacity, particularly for contributors who have programming skills but are still building their C. elegans biology knowledge. So this is interesting, kind of these pushes. And this is basically what we've been doing for, quite a while. There's always been this sort of project management push within OpenWorm. I think a lot of open source organizations do this, where you want to do the things you want to do, you want to build things, but you also have to have sort of a project management push as well. And so it's like thinking about how to connect people within the project, how to connect them to resources, and managing institutional knowledge. So that's kind of the thing that was pointed out here. There's also the Connectome toolbox. This is something that Porrig Gleason has worked on with his students. He has a lab at University College London, and he's worked on this Connectome toolbox for a couple years now. They've been working on building software in Python. So they're working on this unified Python API for assessing more than 30 C. elegans connectome datasets. As I said, since White in 1986, there have been many new revised datasets for different types of instances of the connectome and so forth. So there are actually 30 that they've identified from White 1986 through Wang 2024. So there are all sorts of, if you go into the literature, you'll find different proposed connectomes and collected in different ways, maybe for males, maybe for hermaphrodites, etc. It is foundational infrastructure across many of the design documents, and a paper describing the toolbox is nearing completion. So this is, I think I talked about the connectome toolbox in a previous meeting, but this is, of course, this is the repository for it. Basically, you have this live site, which is where all the action is. That's the code in the repository. This is just showing this different ways to plot the connectome, different things that these different data sets have focused on, and how we can use that to build, kind of interrogate the connectome. So if we want to ask scientific questions about the connectome, we know what data set to go to. We know what kind of visualization might be most effective in answering or visualizing the question. And basically, if you're interested in, say, like movement behavior, you would pick one visualization that would be helpful, versus kind of like bulk connectivity, you might pick another visualization. So these are all things that, if you're using the OpenWorm tools for different purposes, you can get what you need out of it, basically. So that is the Connectome toolbox. Then, of course, the funds will be used in consultation with John. The donation will support two things: updating the worm browser, which, of course, is off of the main OpenWorm website. This is the 3D browser that we have with the, where you go in and you can look at the different levels of anatomy, from muscles to the cuticle to the nerves of the nervous system and so forth. And you can explore that and gain insight from that. And of course, now, we're able to update the worm browser with newer types of data, because the original worm browser, I think, was based on data from about 2014, 2015. And so we can update that accordingly. So we can update that with data from larval worms. We can update that from data from dower worms and so forth. Dower is a specific developmental stage where it's between L2 and L4, where the worms grow this very thick cuticle, and they're sort of they I don't know if it's you could call it a hibernation state, but it's something like it's very different from the worms, say in the L3 stage of development. So that's one step, is to update the browser, and then of course student travel awards for those presenting OpenWorm related work at conferences. Okay, so yeah, they've had a lot of different ways to communicate as a community, and this is kind of talks about that and how those have changed, and then a final thank you to John. So thank you, John White, for contributing to OpenWorm in that way. Okay, Morgan.
Yeah, just wondering, um, how we can kind of follow along better with the greater OpenWorm project in terms of like what they're doing with the browser, or how they're getting more data sets and things like that.
Yeah, yeah, I think that's good. Yeah, it's always hard because we're kind of separate in our processes and our organization. But yeah, I think it's always good. Every year I go to the OpenWorm annual meeting, and we kind of go over some of the stuff. And it's always interesting to see what people are up to.
Sure. I mean, is that like best done? I mean, I see a lot of activity, like their GitHub is connected to their Slack, right?
Yeah, yeah.
So it's like I see that activity. I mean, is GitHub the best way to follow along, or like do they have meetings that like weekly meetings or anything?
Yeah, unfortunately, they don't have like unified weekly meetings anymore. They used to do that like earlier in OpenWorm's
Mm,
history. And that's effective in certain ways and ineffective in other ways. I think
mm,
following the GitHub, or following like they have of course the Slack, where you get like any GitHub activity goes to the Slack, and you can like look at it as like a series of pushes or a series of pull requests or whatever. And it's then you can actually see what people are contributing, and it gives you some clue into what people are actively doing. But yeah, it's a little hard to get, because there's so many different projects, to get a sense of what people are doing in another project. And I've kind of, in the past, I've invited people to come in and talk to DevoWorm, and because we're the part of the project where we have active open meetings. But it is challenging to know.
I mean, that sounds like. I mean, that could also be an opportunity
Yeah, I
in
know.
the sense of
Yeah,
like that's something that is needed, or that is something that could actually get more people, um, by doing something to help you know um, track the project better, or by starting a weekly meeting or something like that, like could actually be something to get more people involved, or you know
yeah,
I don't know.
yeah. No, I know what you mean. It's how do you bridge all this stuff going on?
Yeah. Well, I mean, and then the other thing being that, like, I'm sure that there's people involved who would have so much like um, knowledge, and know what is needed, that would be really valuable in terms of kind of navigating um, what data sets would be useful, like what are issues that aren't necessarily on like mentioned on GitHub, but would be great for for the future and things like that. Because there's people like I mean, I just had this like imperial student like trying to find funds to join Ed Boyden's lab, and because like Boyden and Conrad Courting,
Yeah.
right, have been trying to do this like more, very behavioral, um, big behavioral
Right.
projects on C. elegans, um, and for instance,
Yeah.
and again, it's just like obviously really well, um, um, but like very aligned with OpenWorm in terms of what they when I see the presentations, I'm like, okay, this sounds just like OpenWorm.
Yeah.
like um anyway, it's like if you've got any advice on that, or what we could be doing more of, I'd love to hear it. And yeah, I gotta find. Let me dig up that guy's name again.
Okay.
Um,
Yeah.
it's yeah.
Yeah. Right. Right. So I think we're still technically in the Google Summer of Code community period. So again, we're working on that partially in the open source meetings, but I kind of want to encourage people to explore OpenWorm as part of that community period. Think about like what other things in OpenWorm are relevant to what you're working on. So I know that Barshan is our student for this year, and he's taken an interest in um, some of the things that are going on more broadly in OpenWorm. So it may be the case that his project connects to some of those other things. And so, it's just kind of worth thinking about some of those community resources and how they fit into his project, and even some of the people pursuing projects, you know, uh, that are funded directly by Google Summer of Code. I suggest that you go and explore what OpenWorm has in terms of existing infrastructure and expertise. Okay, so anyone else? Did Mehul or Jess or Morgan want to give an update?
No update, Bradley.
Okay.
Just I wanted to tell something. So like I think couple of weeks back, so 3 weeks back, we got the access of data. Like thank you. We signed the documents for it.
Yeah.
So we have just published a manuscript on it. I'll share the link to it.
Okay.
I don't know what will happen with it. It's related to OpenClaw. So this we are just doing for trying different things. Like I don't see anything happening with it. It is like archive, but for AI agents.
Okay. So this is like the OpenClaw repository or something. I've seen this referenced in places. Yeah,
Yes. So it's
ClawRxiv.
archive.
Yeah. Okay, OrthoRL, a 24 reinforcement learning environment for orthodontic aligner staging, version 2 diagnostic update. So yeah, this is on the ClawRxiv. Yeah, so this looks like, this is just kind of going through the paper, Y version 2 or V2, what V2 adds, and then results. Okay, and so this gives some results, and then yeah, looks like reproducibility, conclusion, acknowledgements. Yeah, so this was OpenEnv Hackathon India, OpenEnv Hackathon India. Theme 3.1 world modeling. So this is actually thought of as a world model.
Yeah, that was the category it was fitting in.
Okay, yeah. And yeah, so that looks really interesting. So you had the hackathon, and this was kind of the paper that you've produced. So you have the code at
Yeah,
the end, the skill file. You have
yeah.
the skill file it's not code. It's sort of is this like the set of prompts plus the code, or what is this?
Yeah, it's a setup for agent. Like the agent, once it reads this skill file, it would be able to reproduce this. That is the goal. Like the goal is for the agents to run it, not as like human.
Okay, so it'd be just the steps that the agent would need to do it.
Yes.
Okay, and then
So one,
oh, go ahead.
yeah. So one small issue happened. Like if you see in this V2 version, there is a previous version link also. Like in the um, um, abstract, you can see even down there is like we are mentioning. So this was a mistake. Like we originally wrote a previous version, and we were supposed to update that, but somehow we forgot the API key for it, so we couldn't update it, and therefore we referenced it that this is the second update.
Oh, okay. Yeah. All
Just
right.
some happy that we weren't actually doing it. We were just managing the agents, like 5 agents in parallel, and it was doing something. We were just like, okay, this is interesting, new direction.
Yeah.
Something came up.
So yeah, this is the peer review, AI peer review.
Yeah.
So is this like from an agent as well, or is this?
Yes, a lot of weaknesses.
Oh, okay. So it's like they generate something and then the agents critique it and
Yeah. Yeah.
okay. So yeah, so this is like strengths, grounds the environment in a substantial real world data set, addresses the complex under researched problem of sequential staging, implements a rigorous evaluation framework, provides an honest analysis of a negative result. And then weakness as the primary reinforcement learning experiment is a total failure. Contains several likely hallucinated or future dated citations. Uh, the clinical knowledge head results are statistically insignificant. The reward function design is fundamentally flawed for reinforcement learning. The paper is framed as a V2 diagnostic update. Yeah, and so it just kind of goes through some of the aspects of it. Yeah, it's fine. Yeah, it looks interesting. So, what do you plan to do with it? I guess it didn't work out as you'd hoped, or did it?
So we did not really had any plans. This is the first agent conference which is named as Claw. So we just wanted to be part of it, like see how things evolve in future and stay connected. So this is like first of its kind conference, and we just give it a. Even the reviewer agent are not so good. Like on Reddit, there were a lot of posts that the reviewer agent is hallucinating at multiple points.
Yeah.
So very new. So we just wanted to get our hands dirty.
Right. Yeah. The experience and see how you can, yeah, use. I mean, it's a lot of experimentation right now. I mean, Morgan is doing this fellowship on agentive coding, and I produced a paper recently on agentive coding where I was interrogating the agent and asking it very pointed questions, and then putting together different models from that, and so that's all kind of I think a lot of people are experimenting with that, and so yeah.
Yeah. Can I find the link to that paper?
Yeah,
If
I'll
it
put
is
it in
still
the Slack actually because I talked about it in one of our meetings too. So
okay, I'll see it.
right. All right. So yeah, I don't know if anyone else had any updates before I move on to another feature.
Yeah, just I'm still refining the what had started as the hypergraph library benchmark. Just been adding to that, and like yeah, and trying to. I'm seeing now that even though there's a couple groups that have replicated anthrobots, um, nobody's actually replicated xenobots, or at least not that I've found. And yeah, so just thinking about what kind of, again, we'd love to be doing something at Biopunk that, you know, like, can we use the wet lab to actually do some interesting science that and you know, Levin's work in particular would be um, you know, I think is interestingly accessible as well as you know, kind of frontier, you know. So trying to put together resources and it will um, you know, and have some sort of just like summary white paper on that in terms of you know, if that's something accessible. So
Yeah.
yeah.
So, yeah.
Try,
What's the difference between an anthrobot and a xenobot? Remind me.
oh, so Xenobots are using Xenopus, you
Okay.
know, as you know, the Anthrobots are actually you know, human epithelial cells.
Oh yeah.
So yeah, and like all of these um so I was kind of like trying to take some of what anatomical compiler work and trying to apply it to Xenopus in terms of like, you know, are there things we could do that, for instance, using Davies' synthetic morphology kind of, you know, framework for how to kind of like what are the main classes of modules, how to kind of direct the the form of the organism in ways that would be more xenobot, or you know, like we could you know, like obviously one is just working on the ciliated the ciliated features, but you know, like this is one of the areas where they were actually doing simulations to make a biobot prediction, right?
Yeah.
And you know, like, could you do something similar?
Right.
But like, you know, stick with Xenopus. That's easier and less expensive for us to work with. And you know, but still have something useful, or you know, like still be working on like, can you control form and with form get or make function predictions, right?
Yeah, that's good. Yeah, I look forward to seeing more on that and how that comes out, because I know a lot of the work is, you know, the key is in replicating it. It isn't just in proposing it. So like a lot of people
Yeah.
propose things.
Yeah. So I want to check. I mean, so I want to check in on Kriegeman's. You know, so it was like the postdoc that was working on this. He's got a new lab at Northwestern, and you know, seems to be focused on continuing this. So, like, I think his website is xenopus.group.
Okay.
Or maybe it's like xenobot.group or something. Anyway, um, uh, and um, and then yeah, two groups have supposedly replicated the anthrobots, which is nice, you know, because this is one of the complaints that people have had, or at least, you know, one of the complaints I've heard from some is like, you know, Levin does these experiments and then nobody actually checks them, or nobody's able to replicate them, or something. You know, and uh, I just wonder how much it's just like nobody sees. I mean, they're not very um, you know, I just remember this one group saying like, well, if Levin's work is so good, then why has he got no successful spin-off companies? I was like, I'm not sure that's the measure that you think it is.
Yeah.
I sort of, like, anyway.
Right.
I don't know if like, would you say that George Church has had a lot of good spin-off companies?
Well, yeah, there are a couple of exceptions, but for the most,
well, some of those are spectacularly unsuccessful. Is what I was gonna say. I was just like, sometimes people can just suck up a lot of VC money and actually, make a lot of headlines, but they don't actually make anything. Anyway, yeah. So I'll post it. Biopunk doesn't have an org, but I want to. I'm gonna post this on make an org and post this and see if we can get some actual students working on this.
Yeah, that would be great. Sounds great. Yeah, I think the things about well, a lot of biology experiments aren't usually in the realm of I mean, they're supposed to be replicable, but it's what's the sort of the baseline for replication, or what is the standard? So a lot of times it's you have to have a dedicated lab, and you have to follow protocol just right, and and then of course with a lot of molecular biology experiments, they work in the hands of certain people, or sometimes they work and they don't work in other when you try to replicate it, because of many factors that we don't. And so that's that, and you can't. But with science, you can't necessarily automatically just create a process where you can reliably replicate it in the engineering sense, right? So you in engineering, we create a proof of concept and then scale that up. And in biology, oftentimes we're not doing that. We're trying to find knowledge. We do an experiment, it works, and it might be replicable, but the results are kind of what matters. But also, we'd like to replicate it if possible. And if we have the tool, if we can do that, that would be great. But it's not I think there's been way too much emphasis put on replication as a standard of success or failure, because a lot of times we'll do things and we know that they work because of other evidence. We know, for example, that something should work. Sometimes we do experiments because they should work, right? We don't just do experiments out of thin air. We kind of come to the conclusion that this experiment should be done because these other things have been shown to be the case. So let's do this. And if we can get a positive result, then that supports the idea. And if you can replicate it, great. If not, well, we got it to work once. So, and so that's a fundamentally different view of how to proceed, and I think, people focus way too much on kind of the engineering slash VC approach to this, which is to say, you have to have everything standardized and these sort of bricks that you stack together. And if you're not doing that, you're just kind of engaged in failure. That's not really the way to view a lot of developmental biology. You think even today, where we have a lot of tools that we can use to manipulate things and standardize things, that we still don't have. We're not even close to the engineering slash VC vision there.
Yeah. I mean, there was definitely a certain amount of, I think people are still working on getting their reporting good enough so that there was a reproducibility problem, just because there was kind of a lack of proper documentation,
right?
So what you see in terms of groups really trying to share protocols more completely.
Yeah. Well,
I'm trying to find a diplomatic way of but you know what I'm talking about.
Right. Oh,
Like,
yeah.
like that was a kind of a real problem. And,
Oh, yeah. Yeah.
and I think, I also think of um, that Marilyn Lancaster talk. She gave something that was like, it wasn't a formal, it wasn't an academic talk. It was kind of like a showing off to like Cambridge research.
Hmm.
But she had a, a a section of it devoted to just why we're not able to replicate each other's organized research. like like different groups are not able to replicate other groups' work,
Right.
and like what's missing. um and it did seem to be that like there's actually just a lot of there's a lot of methodological differences that still aren't really being reported.
Yeah.
um, but yeah, it's the translational and the kind of like like this like every PI needs to be a startup founder is not a direction that I think is helpful.
Yeah.
Like if anything, I feel like that makes the reporting like seem like a bigger burden,
Right.
or like a not a burden, but like you're potentially giving away the secret sauce. It's like, yeah, we just had a group come through, saying again, these are biotech startups, and they're just basically telling everybody in the lab, like, don't publish because because agents will just pick up your work, and you know um,
Yeah.
yeah, and it's just like I'm listening to it, and just like I don't want to. Yeah, I was not exactly happy with that particular. Uh, yeah, there.
Yeah.
You can see the tension there.
Right.
Yeah, there.
All right. Yeah. Thank you. Yeah. So it looks like Susan's here. Hello, Susan. What's going on with you?
Sorry, I'm late. It's a holiday here and I kind of forgot. So, um, what's going on with me? I'm just fighting to get some programs as a student. I'm re registered as a student, so I hopefully can get um, Rhinoceros for $90 US, and I can get um, what is it? My simulation program that I want, ANSYS. I'm trying to download ANSYS and get it as a student because I can get it for free. And I promised the fellow that I was writing, supposed to be writing a paper with, that I would do uh, my model in ANSYS.
Oh, okay.
And there's lots of support for it. And there's lots of training programs for it. Like, I should be able to do this, but I now have ANSYS on my computer and I can't get it to start. Why? Because they don't know that I'm a student or something. Not sure. But anyway, so I'll have a fight with them tomorrow since today is a holiday, and yeah, I call it fighting with my computer.
Yeah. No.
I have several things that aren't working.
Well, yeah. Thanks for the update.
I don't know what to say.
Yeah. Yeah. Software is always something. trying to get it to work.
Yeah. But it would be nice to make that my model in ANSYS, because then the professor I'm supposed to be working with might decide that he could actually step in and help with this. Because I've written a summary of my thesis, and I need to put something else with it.
Yeah.
Yeah.
All right. Yeah, so thank you. Good luck.
Thank you.
Okay, so Jesse, before we move on here, Jesse had some news here. Outside of big news about Canvas breaches and stuff, oh, isn't that true? The truth. It's interesting to see the space of AI plus organizational development and transition, and how folks hope it will go smoothly. Did you want to say something about that, Jesse?
We don't have to get into that,
Yeah,
but I mean, I'm doing things in like, you know, like technology change and adoption in and of itself, but I like I think Ethan Malik is the person that I'll cite here. He's pretty on the note, in depth, in interesting spaces, and having interesting conversations where he's at, and he shares a lot of ideas and thoughts. And there was something he cited, like a conversation with someone from, I think, OpenAI about, this discussion about their whole forward deployed engineers stuff and consulting, getting people to adopt stuff. But I think one of the takeaways was, when the rubber meets the roads of trying to implement technology and actual organizations to do things, like, it's nice to have your agent that can manipulate the bits and the data. I think it comes back to the manipulating digital content and bits and data information versus doing things or making things, and just how much that can be a difference between those things. And I'm generally keeping agnostic about any predictions or whatever else on this front, but I just think there's this a kind of an interesting gap between, like Susan's story is very much mirrored to on other people in my life who are starting their jobs or internships or practicums, or they're joining organizations, and as much as there's all this technology and AI stuff, they're dealing with terrible onboarding and questionable registration, and it's just like it's not like this panacea, and it's like more AI may not help it.
right.
So that's a separate topic for another time. We'll have more conversation maybe it's Friday about those things. But just as someone who's both like in the nonprofit space, in the education space, and in industry, looking at people trying to make these big, how much AI do we get decisions, like, I don't know, like it's quite a humbling, let's say, bottleneck that's causing quite a lot of pain points, and even when they be like, oh, well, we can just spin up some app or a company that will help with those pain points. It's like, well, there's still organizational strategy and onboarding and doing things with people that matter. So that's my little side rant. We can go back to regular scheduled programming.
Oh yeah, that's good. Yeah, thanks for sharing that. And I think always have to keep in mind, like, kind of people's experiences with things and how it matches, say, like the hype or the broader kind of gist of what people want to do. So let's go to this paper here. This is Intelligent Soft Matter Towards Embodied Intelligence. This is a paper that is a bunch of authors who, some of whom are familiar names, some are not. Vladimir Baulin is actually affiliated with the Active Inference Institute. He does things with material science and soft matter, but also from this active inference perspective. So I wanted to present this paper as sort of a representation of that viewpoint. So let's get into the abstract here. So intelligent soft matter lies at the intersection of materials science, physics, and cognitive science. So this is again like we talk about soft matter, soft active materials. I think the last meeting we had a paper where they laid out a vision for sort of the interface between intelligent or not intelligent matter, but like adaptive matter and um, developmental biology and bioengineering and all of that. This is adding this cognitive dimension or this intelligent dimension. So, of course, you're dealing with material science and physics, but also cognitive science. And so, this is a field called intelligent soft matter, which promises to change how we design and interact with materials. This transformative field aims to create materials with lifelike capabilities such as perception, learning, memory, and adaptive behavior. Unlike traditional materials, which typically perform static or predefined functions, intelligent soft matter can dynamically interact with its environment. So this is just this adaptive aspect, but it's a little bit more than that because you're leveraging learning and memory and adaptive behavior and perception. We saw examples of this in the last meeting that we had. We had that paper where they talked about you could think of it as maybe sort of aspirational in some senses because you had materials that were augmented maybe by computation or by other types of things. And in this case, kind of they're moving in that direction where they're like perception capabilities. There's adaptive behavior that can be steered and things like that. And so the goal, though, is to dynamically interact with the environment, integrate multiple sensory inputs, retain past experiences, and make decisions to optimize its responses. So they're kind of getting around this idea of materials as being this inert thing into sort of maybe the way tissues behave, or even the way maybe a brain would handle a lot of this same information. And so they're getting around the sort of the traditional view of matter or of um, some sort of material. And so this maybe involves integrating sensors or integrating computation into the mix. But nevertheless, this is treating this as a system is kind of where they're going with this. So the idea is to leverage the inherent properties of soft matter, such as flexibility, adaptability, and responsiveness, to perform functions that mimic cognitive processes. And of course, Michael Levin and colleagues have proposed this idea of basal cognition, which is very similar to this in the sense that there's this sort of the cognitive state that you can generate maybe without a brain, that you can have sort of these cognitive processes that can be mimicked on different substrates. They could be in single cells, they could be in matter or tissues, or they could actually be in brains of different types. And so that's kind of what they're getting at here. By synthesizing current research trends and projecting the revolution, we present a forward-looking perspective on how intelligent soft matter could be constructed with the aim of inspiring innovations in areas such as biomedical devices, adaptive robotics, and beyond. We highlight new pathways for integrating sensing, memory, and actuation with low-power internal operations, and we discuss key challenges in realizing materials that exhibit truly intelligent behavior. These approaches outline a path towards more robust, versatile, and scalable materials that can potentially act, compute, and think through their inherent intrinsic material properties, moving beyond traditional smart technologies that rely on external control. So that's what they're proposing here. So they talk about intelligent soft matter, or ISM, and the aim of this field is to develop materials with advanced capabilities, usually attributed to living organisms, but also now to soft matter. So you can use this for a number of engineering imperatives, a number of different types of scientific applications, etc. ISM holds the promise of enabling a wide range of applications, ranging from sentient nanoscale robotics, capable of navigating complex biological environments, to dynamically adaptive systems that optimize their performance in real time, to materials that not only sense but also actively modify their surroundings, and to neuromorphic platforms for material-based cognition and thermodynamic computing. And so, this is not just creating materials that think, I guess is one way to put it, but to integrate it with existing into existing biological environments. While the initial development of ISM is rooted in the quest to mimic the complexity and efficiency of biological systems, such as neural networks, its potential may go well beyond mere biomimetics. The ultimate vision is to create soft matter agents with fully synthetic intelligence, capable of functionalities that may not even have biological analogs. Similar to macro-scale soft robotics, intelligent soft matter should go significantly beyond passive adaptability towards more sophisticated levels of material intelligence, characterized by emergent behavior and evolving functionalities in response to environmental cues. And then they talk about emergent agency, which arises when the collective interactions of numerous nanoscale components give rise to system-level behaviors that exhibit goal-directed actions or decision-making capabilities that are absent in the individual constituents. So, for example, a self-assembling peptide structure can exhibit dynamic morphological changes in response to localized chemical signals, demonstrating a sophisticated level of responsiveness that can be harnessed for molecular information processing. So that, and then there are other examples such as shape-changing robots and self-healing materials. So this is where this would come in handy. They give specific examples of this vision. So they think about the behavior of active liquid crystals, exhibiting spontaneous flows and pattern formation. Being able, this is usually something that we attribute to stochasticity, but something also we can control using emergent computation. We might be interested in controlling networks of responsive nanoparticles, which of course can sense changes in their surroundings and anticipate future states based on past experiences, and leveraging these abilities through this sort of methodology. Self-assembling polymers can adapt locally for distributed responses in memory, allowing a single detection event to trigger widespread material changes, such as switching from a hydrophobic to a hydrophilic state. So triggering changes in the material, across the material, from local interactions. They also cite dynamic colloidal assemblies, such as magnetic droplets at the air-liquid interface, which form highly ordered patterns that can be precisely controlled using external magnetic fields. And of course, we can use this sort of intelligence to modify this in real time. And then a nanopore interface for higher bandwidth DNA computing can be used for real-time data processing and enhances the throughput of DNA-based computational systems. So there are all sorts of, and they make the analogy here with Maxwell's demon, which is an idea from thermodynamics, which is where there's this thought experiment that was developed by James Clerk Maxwell, in which you had two chambers. The chambers have a little door that opens up. And the idea is that you have, in each chamber, you have two well-mixed sets of molecules. So you have two well-mixed ensembles, and the trapdoor opens and closes in a way that allows for different states of this molecular ensemble to sort themselves, so that over time, one chamber has one state and the other chamber has another state. And this flies in the face of the prediction of thermodynamics, which would be that things should tend to disorder over time. Where if you had, say, like a chamber with two states and you had a trap door, as you open that trap door, there should be a random flow of particles such that there'd be a well mixed ensemble in each chamber after a while. And so Maxwell's demon is basically the inverse of traditional thermodynamic entropy. And so the idea here is to sort of implement Maxwell's demon as this form of intelligent control that can sort of fight thermodynamic entropy and can lead to order from disorder. So that's what this part of the paper talks about. So then, they kind of talk about the main capabilities of intelligent soft matter. They have this figure here, which is largely aspirational, but it basically talks about decision making, actuation, memory and learning, and perception and sensing. And so this mimics sort of a model of intelligence. So for decision making, this is the process of making autonomous decisions based on inputs. Actuation is the capability to perform actions based on internal stimuli. Memory and learning is the capacity to retain information and adapt over time. And perception and sensing is the ability to sense and respond to environmental stimuli. So they actually have a couple of examples here where they talk about, in one case here where they talk about anticipatory state dynamics, they talk about the evolution of the internal state of the material, much like you might have an internal state of an organism that leads to the sort of cognitive behavior. So every agent that exhibits cognitive behavior has some sort of internal state. And that internal state is regulated. Perhaps it's sort of through cognitive processes and information processing. Perhaps it's through some sort of homeostatic mechanism. So this is something that, what are the distinctions between intelligent behavior and homeostatic control? That's an interesting question that they don't really talk about here, but that's relevant. So they talk about anticipatory state dynamics. They bring up the example of viscoelastic materials subjected to repeated stress cycles, which can develop an internal structure, which is a memory encoded in polymer conformations or spring rest lengths that reflects the history of applied loads. So you can have a situation where you have a system that responds to the environment over time such that it develops this sort of memory structure. And of course, that helps the system perform better in the future, given equilibrium conditions. So they also talk about the internal memory state of the slime mold Physarum polycephalum, which has a tubular network hierarchy that responds to nutrient sources and guides future migration towards locations where their nutrient sources exist, embodying a prediction of resource availability based on past encounters. And so they talk about how active matter systems operating near critical points may also exhibit similar anticipatory dynamics, where long-range correlations allow the system to sense impending global changes based on local fluctuations. So they're making this analogy between things like viscoelastic materials and slime mold colonies, and then thinking about how those systems produce these adaptive responses and think about how that can be replicated in matter. There are other things they talk about: physical goal embodiment and error correction. So this brings in the idea of minimizing free energy and homeostatic control. And they don't spend a lot of time on this, but they do talk about how active inference can help here. They say that theoretical frameworks like active inference formalize this, suggesting that biological systems minimize prediction errors relative to an internal generative model representing preferred or physiologically viable states. Okay, so that's that part. Then they kind of get into memory and what they mean by that. And so this is basically the ability to encode and retrieve information based on past interactions, such as deformation patterns or exposure to particular substances. So this is, if you think about things like hydrogels, hydrogels can retain and forget information based on thermal stimulation, showcasing how soft materials can encode and retrieve information based on those past interactions. And so then they talk about self-repair and learning and how self-repair mechanisms enable the material to mend itself and develop resistance to recurring stressors, allowing it to learn from repeated exposure. So self-repair involves learning, involves some encoding of learning as a form of memory. And then, of course, they talk about adaptive pattern recognition, which is where some intelligence is presented with a pattern and it recognizes that pattern through training and similar patterns through sort of recall of the original pattern. This talks about the principles of intelligent soft matter. So this actually resembles the figure that we saw in that paper we talked about in the last meeting that we had. But this is a little bit different. This is just the 4 principles that govern intelligent soft matter. So the first one is self-referential evaluation, where there's some sort of internal assessment of the environment or of the current state and proactive decision making based on what that state is. There's self-organization, which is the spontaneous structure formation that occurs without external templates. So that could be from the interactions of agents or of component parts, and they organize into larger structures. Active inference is part of this as well, which is adapting to minimize prediction error and surprise. The idea basically that you shouldn't have sort of these unanticipated things that occur that the system doesn't know how to respond to. And this happens through minimizing free energy and other types of mechanisms and processes. And then emergent agency, which is collective decision making and goal-directed behavior. So this is where you have your global structure, your collective structure, and it then can make decisions and engage in goal-directed behavior, whereas individual agents may not be able to do that. A lot of interesting, this is the section that kind of defines the concepts here. And then they talk about the challenges and opportunities, which of course are many because this is largely conceptual. But still, you have a lot of, promise here. So this is why they're writing the paper. And this is a figure kind of summarizing these challenges, including scalability, energy management, integration complexity, complex behaviors and cognitive functions, durability and stability, and material complexity. Okay, so that's all I have for that paper. Another very aspirational paper, but I think it's useful for thinking about some of these broader questions as to what we can do with matter and physics and biology.
So I have a very brief question, and I confess I could only hear part of it and had to step away. But I was curious about the title and sort of the conventional what was the end of it, like towards embodied intelligence? Is that what it?
So the title was towards embodied intelligence. That's the way they define intelligent soft matter.
Okay. Do you see? Because I don't know. Like I'm familiar with like Levin's agential materials and things like that, and intelligent soft matters is interesting, and I don't know. Like I was curious, and this is again I've totally missed. So if this is just I'll go back to what I said. You could just say that's fine. But like in their sort of like towards embodied intelligence, are they? I feel like that's something that a lot of people could say about what they're trying to do, but what was there something specific about? Okay, we see intelligent soft matters as an inherent addition or updating or expansion of embodied intelligence, as in a more overall robust thing, or they kind of was it like oriented towards we want to move the thing in this direction, or we're kind of just a tack-on statement that's sort of loosely framing what they're gesturing at. I don't know if that makes sense.
Yeah. Well, I think it's like they're using sort of principles of embodied intelligence in this context. So, intelligent soft matter, of course, it has to be emergent. It has to have a larger structure than just the individual components. So if you're dealing with like polymers or like a collective of polymers, basically, you have it. So it's embodied in a larger structure, and then it's deriving its intelligence from the environment. So it's interacting with the environment. A lot of these things are more, they could be maybe more analogical in that this is representative of what you see in embodied intelligence. So for example, if you're looking at like an embodied robot or an embodied human or an embodied insect, you have those kind of concepts. They come from like analyzing a system, right? So like those things exist. The behaviors are generated through what exists there. And if you're looking at animals, a lot of times you don't actually know what the internal mechanisms or the internal models contain. You're just kind of inferring that. In this case, you're building this from scratch, so you have to define all those things, and you have to attach like familiar concepts. So is it embodied intelligence? Yeah, I guess, but I mean, it's not the same as you would find in like an animal system or even in a robotic system where like you had an embodied robot like a Boston Robotics dog or something, so it's very different, but it's still, I think they're trying to apply the principles of embodiment to what they're doing.
Yeah, that's cool. And I think, as you know, in some of my other messages, I'm asking in part because I'm my question is telling of some of the things that I'm attempting to flesh out in other ways, and I think this paper might actually be a nice. This paper is relevant to some of that in the sense of where people are trying to take, in a really broad sense, like updates to cognitive science or reconsidering what's been sort of bracketed in or bracketed out of considering as they're developing X, Y, or Z. So yeah, good to hear from this. And I guess, is that going to be our last call for today?
Yeah, I think so.
All right. Well, it's good to see everybody, and I'll see you next week.
There was a SLAM meeting.
There was? Okay.
There was a SLAM meeting, and it used circuits for intelligent, adaptive systems. It had an intelligence to it, and it was just a grouping of circuits that they'd come up with, electrical circuits.
Yeah.
So that was interesting, and I know that cells act like this. They adapt. If you stretch them, then they produce more attachments. Like they change their properties. So
Yeah.
they're an intelligent material.
Yeah.
So just wanted to say that. Yeah.
Good. Well, that's great. Yeah. Thank you.
Okay.
Okay. Well, I think that's all for today. Thank you for attending, and see you next week. Bye.
Take care.
Take care. Bye.